FDA staff recommended no. The advisory committee voted yes on all 8 questions, but FDA did not approve BPC-157, KPV, TB-500, or MOTS-c. So what changed? And why did 4 names require 8 decisions? This investigation tests the human evidence, chemical identities, and product quality gaps behind the result. Here is the first answer. The committee recommended 8 bulk substances for the 503A bulks list, a pharmacy compounding pathway. The advice is not final FDA action, and no finished drug was approved for safety and effectiveness. Each name was split into freebase and acetate, related forms, but not automatically evidence equivalent. Some nomination files also failed to align on the name, identifier, and certificate. So the deeper mystery is not simply did the committee vote yes, it is which substance does the evidence actually fit. By the end, you will know what the recommendations change, what they do not change, and which claims survive a direct human evidence check. I'm Sharif El-Gendi. Welcome to Peptide IQ, where we investigate peptide and GLP-1 claims through human evidence, regulatory facts, and product quality science, not hype. This investigation is focused on the 4 substances discussed by the committee on the 23rd of July: BPC-157, KPV, TB-500, and MOTS-c. The official uses under review were much narrower than many online claims: ulcerative colitis for BPC-157, wound healing and inflammatory conditions for KPV, wound healing for TB-500, and obesity and osteoporosis for MOTS-c. This video is for education only and is not medical advice. Do not start, stop, inject, combine, or purchase any peptide or medication based on online content. Speak with a qualified clinician about your own treatment and safety risks. Now we need to answer the question most headlines skip. What was the committee actually deciding? Imagine 2 regulatory doors. One door leads to normal FDA drug approval. That route asks whether a defined drug product has adequate evidence of safety, effectiveness, and manufacturing quality for a specific use. The second door concerns pharmacy compounding. That is the door being discussed here. Under Section 503A, a pharmacist or physician can use certain bulk drug substances only when specific legal conditions are met. One possible route is for the substance to appear on the 503A bulks list. But here is the catch. A place on that list would not transform the resulting compounded product into an FDA-approved medicine. FDA's presentation states that compounded products are not reviewed by the agency for safety, effectiveness, or manufacturing quality before marketing. For this list, the agency balances 4 main questions. Can the substance be physically and chemically characterized? What safety concerns exist? What evidence supports or fails to support effectiveness? And what is known about historical use in compounding? There is another unusual detail. The nominations for these substances were withdrawn, but FDA chose to continue the evaluations on its own initiative. Peptide IQ mini-verdict. This meeting concerns compounding eligibility. It is not a shortcut to drug approval. This is where the identity thread becomes visible. The internet sees 4 peptide names. FDA sees 8 distinct bulk drug substances. Each peptide has a proposed freebase form and an acetate form. They may share the same active peptide component, but they are not automatically the same active pharmaceutical ingredient. Think of them as members of the same chemical family with different passports. The family resemblance may be obvious, but the exact identity still matters. Different forms can have different physical properties, different stability, different solubility, and potentially different pharmacokinetic or pharmacodynamic behavior. That means one form cannot automatically borrow the evidence, quality data, or regulatory conclusion of another. FDA also found inconsistent naming in the nomination packages. In several cases, documents appeared to identify one form, while certificates or chemical information pointed toward another. This is not paperwork for the sake of paperwork. If the name does not reliably identify the substance, the evidence may be attached to the wrong chemical form. Mini-verdict. A common peptide name is not a complete chemical identity. Let's open the first case file. BPC-157 is often described online as the healing peptide. Tendons, ligaments, muscle, the gut, sometimes almost anything that has been injured, but that was not the committee question. FDA evaluated the freebase and acetate forms specifically for ulcerative colitis. There is extensive laboratory and animal research around BPC-157. Some rodent studies report effects involving gastrointestinal injury, fistulas, inflammation, and tissue repair. But mechanism and animal response are not proof of a human treatment outcome. The direct human evidence for ulcerative colitis was much thinner. FDA identified one small exploratory treatment trial using a rectal enema. The publicly available report was a meeting abstract with limited methodological and outcome detail. That makes the result difficult to verify and interpret. FDA also found no ulcerative colitis studies using the proposed oral, subcutaneous, nasal, or transdermal forms. 2 short rectal enema studies provided some human exposure information, but treatment lasted no more than 2 weeks. Safety monitoring was limited, and that cannot establish long-term safety for a chronic condition. FDA also identified unresolved questions involving characterization, impurities, aggregation, and immunogenicity. The agency therefore concluded that the evidence was insufficient to determine effectiveness for ulcerative colitis or adequately characterize safety. Before the committee vote, FDA staff proposed that neither BPC-157 freebase nor BPC-157 acetate should be added to the list. Peptide IQ verdict. The biology is interesting, the direct human evidence is limited, and the broad heals everything claim is unsupported. The second case file begins with a plausible signal and a missing bridge. KPV is a 3-amino acid peptide related to a fragment of alpha-melanocyte-stimulating hormone. In laboratory and animal research, it has shown anti-inflammatory activity. That provides biological plausibility. It does not provide clinical proof. The nomination proposed topical products for wound healing and inflammatory skin conditions, but FDA did not identify clinical studies showing that KPV treated those conditions in humans. The agency also did not identify human exposure data for KPV through any route. There is another practical contradiction. An in vitro study suggested that KPV did not pass efficiently through human skin. That could limit systemic exposure, but it could also limit the ability of a topical product to reach the deeper tissue where an effect is expected. Low penetration is not automatically a safety victory. It may also be an effectiveness problem. FDA found no adequate non-clinical toxicity program. It also found unresolved questions involving impurities, aggregation, microbial testing, and the performance of the finished cream or gel. No adverse event reports were found, but an empty reporting database is not proof of safety, especially when reporting from 503A compounders is incomplete. Before the vote, FDA staff proposed that neither KPV-free base nor KPV acetate should be added. Peptide IQ verdict. Anti-inflammatory biology is preliminary. Human effectiveness and safety for the reviewed uses are unsupported. With TB-500, the identity thread becomes impossible to ignore. Online, TB-500 and thymosin beta-4 are often treated as interchangeable names. They are not the same substance. Full-length thymosin beta-4 contains 43 amino acids. The TB-500 substance evaluated by FDA is a synthetic 7-amino acid fragment from that larger peptide. That distinction changes how the evidence must be read. Studies involving full-length thymosin beta-4 cannot automatically prove that the 7-amino acid TB-500 fragment produces the same outcome. In fact, references submitted with the nomination included research on thymosin beta-4 rather than the specific TB-500 substances under review. For wound healing, FDA did not identify human exposure data involving TB-500 freebase or acetate. It also found no convincing non-clinical in vivo evidence showing that these specific substances promoted wound healing. One laboratory scratch wound experiment did not show wound healing from the intact TB-500 peptide. A smaller metabolite appeared biologically active in that model. But a metabolite signal is not proof that the marketed parent substance heals human injuries. FDA also found no adequate acute, repeat dose, reproductive, genotoxicity or carcinogenicity studies for the reviewed substances. Because proposed products involved injectable use, the agency highlighted unresolved immunogenicity risks involving aggregation and peptide-related impurities. Before the committee vote, FDA staff proposed that neither form should be included. Peptide IQ verdict. The wound healing claim is unsupported, and borrowing full-length thymosin beta-4 evidence for TB-500 is misleading. Which peptide claim needs the clearest follow-up investigation? BPC-157, KPV, TB-500, or MOTS-c? Comment the name below, without sharing doses or suppliers. The fourth case file has the most futuristic story. MOTS-c is a peptide encoded within mitochondrial DNA. It has been linked in research to metabolism, exercise adaptation, muscle function, and aging. The biology is genuinely interesting, but the clinical claim is a different question. For this meeting, the main evaluated uses were obesity and osteoporosis. Laboratory and rodent studies have reported effects involving glucose and lipid metabolism, bone formation, bone breakdown, and vascular calcification. But those findings remained in cells and animal models. FDA did not identify clinical studies in which MOTS-c freebase or acetate was administered to humans for obesity, osteoporosis, or the other nominated uses. That distinction matters because measuring naturally occurring MOTS-c in people is not the same as testing an administered MOTS-c drug product. One laboratory study also found that MOTS-c was rapidly broken into smaller peptides when incubated in human blood. That raises a basic unanswered question. Can an externally administered product produce an active concentration in the human body for long enough to generate the claimed effect? FDA also found no adequate clinical pharmacokinetic data, no human exposure data, and no adequate toxicity program for these forms. No adverse event reports were identified, but again, no reports is not evidence that no adverse events occurred. Before the committee vote, FDA staff proposed that neither MOTS-c freebase nor MOTS-c acetate should be included. Peptide IQ verdict. The mitochondrial biology is promising. The human weight loss and osteoporosis claims remain unsupported. Across all four case files, the same product quality problem kept returning. A familiar peptide name does not answer every question about what is inside a finished product. Think of a certificate of analysis as a passport for one tested sample. A passport may help confirm identity at one checkpoint. It does not inspect every bag traveling with that person. A certificate might report identity or a headline purity result, but that does not automatically establish the full impurity profile. It does not prove that the peptide has not aggregated. It does not establish bacterial endotoxin levels. It does not confirm microbial quality, stability, potency over time, or finished product performance. FDA repeatedly found missing or inadequate information across those areas. The agency also found inconsistent naming and uncertainty over whether some documents referred to a freebase, an acetate salt, or another related form. This does not prove that every product carrying one of these names is counterfeit or contaminated. It means product-specific certainty has not been earned merely because the label looks scientific. Outside a verified and appropriately regulated supply chain, identity, potency, stability, microbial quality, and chain of custody may remain uncertain. Peptide IQ verdict. A peptide name is not a complete quality system. Assuming all products are equivalent is misleading. Before we reveal the vote board, we need a translation key. If the committee votes yes, it is recommending that FDA consider placing that specific bulk drug substance on the 503A bulks list. That does not approve a finished product. It does not prove effectiveness. It does not certify every pharmacy, supplier, or online product. And it does not turn the peptide into an FDA-approved medicine. If the committee votes no, it is recommending against inclusion of that specific substance on the list. That weighs against its eligibility for this particular compounding pathway, but the vote itself is not a normal drug withdrawal. It does not erase laboratory research, and it does not, by itself, answer every question about online sales, enforcement, or possession. The committee advises. FDA makes the final regulatory decision. Any later change to the official bulks list would require further agency action. The advisory vote is important, but it is not the final page of the regulatory story. Mini verdict. Panel recommendation is not drug approval, and panel opposition is not the same as an instant universal ban. Now, the complete verified vote board. As each row appears, watch one thing. Did any acetate form break from its freebase partner? For BPC-157, both forms received 8 yes, 6 no, and 1 abstention. Both were recommended for inclusion. For KPV, the two rows were identical. 8 yes, 6 no, and 1 abstention. Both recommended for inclusion. For TB-500, the same pattern appeared again. 8 yes, 6 no, and 1 abstention. Both recommended for inclusion. MOTS-c changed the numbers, but not the direction. Freebase and acetate each received 7 yes, 5 no, and 2 abstentions. Both were recommended for inclusion. Now the pattern can be revealed. None of the 4 chemical pairs split. All 8 questions received majority support, but none was unanimous. And here is the reversal that matters. Before the meeting, FDA staff proposed non-inclusion for all 8 substances. The advisory committee recommended inclusion for all 8, but that vote did not approve a finished drug, and the vote did not place any substance on the list by itself. These recommendations are advisory. FDA must now decide whether and how to act. Peptide IQ mini verdict. The committee supported a possible 503A compounding pathway. It did not grant drug approval, and it did not strengthen the underlying human evidence. So after the favorable votes, which claims are actually supported? That is the final verdict. So what does the evidence support after separating the vote from the marketing? For BPC-157, animal research is extensive and early human information exists, but the evidence does not establish effectiveness for ulcerative colitis. Verdict: unsupported. For KPV, the anti-inflammatory biology is interesting, but FDA identified no human clinical evidence for the reviewed wound healing and inflammatory uses. Verdict: unsupported. For TB-500, the evidence problem begins with identity. It is not the same substance as full-length thymosin beta-4, and no human effectiveness evidence was identified for the reviewed wound healing claim. Verdict: unsupported. The evidence borrowing is misleading. For MOTS-c, the mitochondrial and metabolic biology is promising, but direct human treatment evidence for obesity and osteoporosis was not identified. Verdict: promising biology, unsupported clinical claims. Across all 4, long-term safety and product-specific quality remain inadequately characterized, and the label cannot prove that 2 products contain the same form, potency, purity, stability, or microbial quality. Science does not always end with a dramatic yes or no. Sometimes the most important conclusion is that the direct human evidence is not yet strong enough. Based on the evidence currently available, the Peptide IQ verdict is clear. Interesting biology, limited human proof, unresolved product quality. If this investigation helped you separate the committee vote from drug approval and the evidence from the marketing, press like, subscribe to Peptide IQ, and share this video with someone following BPC-157, KPV, TB-500, or MOTS-c. But this was only the first day of the FDA peptide review. The committee also examined Semax, the so-called brain peptide marketed for focus, memory, and neurological recovery. Does Semax have stronger human evidence, or is it another case where the mechanism is real, but the certainty is not? That full investigation is on screen now. I'm Sharif El-Jendi. Stay curious. Question the marketing. Always follow the evidence.